So we may as well get into it. So today I'm very happy to announce that we're shared by Ada Hamosh and Joanna Amberger. Did I say your name there correctly,
Yes.
Joanna?
Yep.
Excellent. From John Hopkins, but giving us a refresher as well as an update today about many of the features that are in the OMIM database. So I'll hand over to the two of you.
Thank you. Thanks very much.
Thank
So
you.
I want to preface this by saying I talk too fast. I try to talk slowly, but then I accelerate because I get excited. So if I'm it's gobbledygook, could somebody say, to slow down. And the other thing is, if anything is unclear as I go, please feel free to interrupt, ask your questions. It'll be much more effective that way because if you're confused about something, I want to make sure you understand it. So I am going to make this smaller. And then I'm going to proceed. Okay, so in the beginning, there was Mendel and his piece, and then there was Victor McCusick, who started life as a dairy farmer, son of a dairy farmer, but then became a physician and was initially a cardiologist, and then developed an interest in genetic disorders of the heart, and then expanded to everything else. And he was an inveterate note taker. So, and he had all these little brown books, and he took notes. And the first book he wrote was about the disorders on the X chromosome or X-linked. Well, we didn't know anything about anything yet, but about X-linked disorders. And then gradually he started writing the first edition of OMIM, which was this. This is his official portrait, and I love it because it shows the books behind him, a picture of the Amish Madonna. He spent a lot of time among the Amish, learning about recessive disorders, and then heritable disorders of connective tissue. So he wrote 12 editions of this book, and until this edition right here, he wrote them all by himself. And well, with lots of help from editors, but he wrote them all by himself. And it was Mendelian Inheritance in Man. And it was initially called a catalog of autosomal dominant, autosomal recessive, and X-linked disorders, and ultimately a catalog of human genes and genetic disorders. First edition in 1966 with 1,600 entries, and the last edition in 1998, printed edition 1998, with 8,400 entries. And you can see it's 3 volumes, and that's onion skin paper. So if we tried to publish it now with over 27,000 entries, you wouldn't have a shelf big enough to hold it, I don't think. So, and Mim was always on computers. And actually, that is why all OMIM titles are in all caps, because in 1966, the only way to type for computer cards was in capital letters. And it makes some people crazy that the titles are all in caps, but it's actually, I find it very useful because then I know the source is OMIM. Like, oh, let me not go look where this comes from. I know where it comes from. So, and it was part of a information retrieval experiment project at National Library of Medicine, which supported full text retrieval using natural language as far back as before most of you were born, 1987. MIM went online and became OMIM. And it was made available on the internet from the Welch Medical Library. And to give you a context, those servers, which are now, I don't know, probably under 1,000, maybe under 100, Joanna will correct me, cost $20,000 in 1987. So you can imagine what that would be today. In 1995, OMIM was moved from the World Wide Web onto the World Wide Web by NCBI and was incorporated into their Entree suite of databases. It is still incorporated into the Entree suite, but it is best used within omim.org, which debuted in January of 2011. Okay. So this is the growth of the different volumes, and the last published is MIM 12. And then these are just some standard dates from different times to show that we keep working. And as of today, there are 27,791, I think it's two today actually, entries, and we add about 250 new phenotypes per year. So this is the information framework within OMIM. There are phenotype entries, and phenotype entries have copious external links. A link exists in an entry only if there is a page on that subject in the resource to which we are linked. So if there isn't any information about, I'm going to pick a phenotype like cystic fibrosis in some other resources, there won't be a link. If there are excellent specific information in other links, it may only be in the cystic fibrosis page. A phenotype entry also is where you would find a clinical synopsis, which is a link out into a structured information about the phenotype. There are gene entries, and all variants, allelic variants in OMIM live in a gene entry because that's where the variants are. They're in the gene. And then similarly, gene entries, each gene entry has specific external links that are topical to the other resource. The relationship between phenotypes and genes live within the gene map, and there's information within the gene map that is organized into phenotypic series and molecular series. I'm going to tell you more about both of those in a minute, and then can be visualized through the PhenoGene graphics. There are dotted lines because not every, but most phenotype entries have a clinical synopsis. Not every phenotype has a known gene. And then whether they come out into the phenotypic series and molecular series depends on us because each of those is manually curated by us. Okay, and we'll tell you who us is in a minute. So in OMIM entries, each entry has a unique stable MIM number, which are heavily used in the biomedical literature, and they cross-link with other data resources, and I'll spend a little time talking about that in a moment. Primary titles and other aliases and symbols, including formerlies. So nothing ever leaves OMIM. Something may not be a word or a name you should be using anymore, but it'll still be there because if you go back in the literature, you may find the older name and you're like, well, is that the same? Is it something different? What does it mean? If it's the same, that name is still going to be there and findable. And MIM entries are rich full text descriptions of genes and genetic phenotypes and disorders. And they have a structured format that is both human searchable, which we'll spend a little time on, but also importantly, computationally searchable from the back end. Entries in OMIM are expert curated with predictable semantic structure, and every single thing you find in OMIM is cited to the peer-reviewed biomedical literature. This is the tiny group of human beings right here who work on OMIM every day and produce what hopefully you guys are using. There's lots of other people in the past who have written for OMIM at various times, for MIM and for OMIM at various times, but this is the staff right now. Okay, and they work very hard, and I thank them very much for everything they do. This is the growth of gene phenotype relationships as of yesterday, or I guess 2 days ago. And you can see in the beginning there were just phenotypes, and then over time, some more and more genes with phenotype causing mutations were identified. There are always more phenotypes than with a known molecular basis than there are genes that cause them, because a third of genes, of disease genes, have more than one disease associated with them. And we could talk about lumping and splitting later. Okay, so here's the OMIM homepage, which hopefully you're all familiar with. This is omim.org, and this is the search box, and type what makes you happy. So in terms of genes, I'm going to go through genes and then phenotypes and then other information. There are over 17,570 gene entries in OMIM. They include obviously protein coding genes that may be transcription factors, microRNAs, long non-coding RNAs, locus control regions, and more. If it's a functional element of the genome, we call it a gene. Over 5,340 entries have allelic variants, and over 35,000 allelic variants exist in OMIM. So our allelic variants, I'll get into this a little bit more later, are sample variants, we're not a variant resource, as you all know. Okay, so now we're going to do the anatomy of a gene entry. So I have picked fibrillin 1 because I like it. The top line, this is the MIM number for fibrillin 1. This asterisk right here indicates that this entry describes a gene. This one doesn't have a lot of alternative symbols, but here they are. Usually the official gene symbol is the same symbol that we use, but if for any reason it's not, the HGNC approved gene symbol is always right there with a live link to that resource. There are cytogenetic locations and genomic coordinates. And then at the top of the table, at the top of the entry will be a gene phenotype relationship table if there is one, right? So in this case, there are 8 different autosomal dominant conditions caused by this gene. And if you click here, you will see a table of those phenotypes. And these are the side-by-side clinical synopses of these disorders. And this is just the top. So I'm starting with the title, the gene, the inheritance, the growth, head and neck, and then obviously this goes on at this size for pages and pages, right? I'm not going to show them all to you. But the thing that's important to see here is you can see that in Marfan syndrome, people are tall. And in Marfanoid progeria lipodystrophy syndrome, people are tall. But in these guys, this is severe short stature. This says short stature, this says short stature, and this says short stature, which would mean that you cannot say FBN1 related disorder because you would be talking about tall people and short people as if it's one thing, and it's clearly not. Um, so so that's, you can see all of them side by side. You can do this in any gene entry. So the next thing I'm going to show you is this table view of the allelic variants. Okay, so we're going to click on that. And you can see that there are 70 selected examples. You could click here to find out what selected means, and I'm going to show you in a slide in a minute. And then these are the different phenotypes, and obviously this is just the top 17, so there's more to go. But 70 doesn't seem like enough for a gene known for as long as FBN1 has, and the many different phenotypes. So you can click on all ClinVar variants, which takes you straight to ClinVar, and you can scroll through to see, and you can see that there's literally thousands more. So you must go to another resource to get all the variants and to look up variants. You all know what ClinVar is, so I'll skip that slide. But this is an example. In this case, I've gone up to one of the variants, and you can see that it's pathogenic, and it's got 2 stars because 3 out of 3 submissions agree. And you all know this is when it was first described. This is when it was last submitted, last formally evaluated. And then as we go on, these are the calls. This is, you can see a standard lecture that I give to everybody, whether you're a biocurator or a third-year medical student. But here you go. You might as well know it all. And here are the details that you can find out more. Okay, so now I want to just tell you about PhenGene graphics. I don't know if any of you use this, but I want to just let you know a little bit about this. So this little question mark, if you click on that, it gives you a static PDF that explains what each of the things that you see, what the colors mean, and what it means when you toggle on or off. So that's just for your information. The next slide shows you the fibrillin 1 gene phen graphic. So in this case, we've picked fibrillin 1, and you can see that these are the 8 phenotypes that are associated with fibrillin 1. And you can see that in for geliophasic dysplasia, I've gone to that phenotypic series. There are 2 components of the 2 other components of the geliophasic dysplasia phenotypic series caused by ADAM TSL2 and LTBP3. So you're basically going in and out from gene to phenotype to gene to phenotype, and here you have phenotypic series as well. So it's a way to just, it's all information that's in the entries, in the tables, it's all the words in OMIM, but if you're a person who likes to visualize data, this is one way to do it. The other way to do it, that was called the linear view, and no hierarchy is implied. This is just relationships, and you can click on any of these to reorient this whole picture based on that particular choice. This is, and I did not make this into a video, but this is a radial view, so it rotates. And here it's centered on FBN1, but if I clicked here, I'd go to Marfan lipodystrophy syndrome. If I clicked here, I'd go into the progeroid syndrome series, phenotypic series, etc. Okay, so the other thing I want to show you here is that right up here, there's this little hidden thing called ICD plus, and it might be important for you if you're trying to figure things out. So these are all the codes that are for, in theory, exactly the same condition, or hopefully not theory, really the same condition. So this is the MIM number for Marfan syndrome, and these are the SNOMED CT codes, ICD-10-CM codes, ICD-9 codes, ORFA codes, disease ontology, and Mondo codes are all right there. And that should be true of any OMIM phenotype that's in any other resource will have that right there for you to look at. Okay. And this was the example, and I didn't show this before, of the topical links. So it's always toggled to clinical resource, and where there's an arrow, there's more information below it. But of course, all of these have information as well. And on the left, the table of contents just takes you directly to that part of the entry if you want to jump to some other segment. Okay, so now I'm sorry, I didn't click it, but I've just gone to a clinical synopsis. I think you all know where that is, but I'll go back a second. I've clicked the clinical synopsis. So this is the clinical synopsis as a single page for Marfan syndrome. And in this case, this is again, I'm at the top of it. And what I want to show you here is where there is a little bust icon. If you mouse over that, you see a picture of that feature. Okay. So this isn't a patient with Marfan syndrome. This is a picture of dolichocephaly. And if you click here on elements of morphology, it'll take you to that page, and which includes maybe, hello, there, the definition of what it is, etc., and sometimes has additional pictures. Okay. Any question questions about this before I go further?
Autumn, I just wanted to mention
Yeah.
that the MIM number prefixes,
Yeah,
because the ICD codes will only be available in phenotype entries, not in gene entries.
right.
And those are denoted by either a pound sign or a percent sign. Genes have an asterisk.
So that's what she's talking about right here. And then in this case, what I've done is I just toggled feature IDs up here. So if for any reason you're, itching to know the ID, either, the UMLS ID, the HPO ID, some of them have SNOMED CT codes. For these particular features, you can find them here. Obviously, if you're doing a bigger search, it's easier to go through the back end through the API. But in any case, it's all right here. Um,
And then
so
there was just one question related to that
yes,
in the chat as
please.
well. So is in the clinical synopsis, is it connected to HPO or is it independently maintained?
It is completely independently maintained. We create our own clinical synopses, and it's off of the papers that are included in OMIM. If they aren't, if we didn't just, we get the information on patients with mutations in the gene. Okay. And
Yeah,
but
that's where we only include features from patients reported with mutations in the gene. So if a uh, existing phenotype is in and we then find a gene associated with it, we will revise the clinical synopsis to be based on those patients with mutation in the gene.
okay. Um, let's see. Sorry, I need to, there we go. Okay. So in this case, I want to show you a couple more features in case you want them. So in this case, I have typed in the word progeria, and this is an old search. It may not be completely up to date, but in any case, I typed in the word progeria and I got back results that include phenotypes, but also genes, right? So I have kind of both here. So if I want to only see the phenotypes that have progeria, I can click clinical synopsis right here and I get this view. This is called we call it, who cares, the clinical synopsis quick view. And what it shows you is the phenotypes. And then underneath in green are the headings from the clinical synopsis. And I can, if I mouse over them, I get the list of the features within that heading. Okay. So it's another way to look at the information other than side by side, but most people like side by side because you can look at everything up and down quickly, and you can do that as this. So in this case, I've done the same search and I've clicked these two and I said compare selected, and then I'm looking at these two side by side. This is a particularly useful thing to do in clinic. If you're trying to figure, could it be this, could it be that, you can just see the features side by side. Okay. So what kind of phenotypes do we have in OMIM? As you can imagine, we have single gene Mendelian disorders, obviously. We also have phenotypic traits. So hair and eye color, PTC tasting, things that are clearly inherited in Mendelian fashion. They don't necessarily have to give you a disease. It just is a phenotypic trait. Susceptibility to drug reactions, things like malignant hyperthermia, warfarin sensitivity, other drug reactions. Altered susceptibility or reaction to infection. For example, herpes simplex encephalitis, which is based on, recessive mutations in 4 or 5 different genes. And it's a combination of having that and exposure to herpes at the wrong time. Progression of HIV infection to AIDS or protection from that. Those are all Mendelian phenotypes. Germline susceptibility to cancers, for example, BRCA1 and 2 in breast and ovarian cancer. We are not the somatic acquired cancer resource at all. And then we also report recurrent deletion and duplication syndromes, some of which may have actually been recognized as a Mendelian phenotype before it was known that the etiology was a chromosomal deletion or duplication. We do not include one-off deletion and duplications. Only they have to be recurrent, and we don't do all recurrence either, just where you have to. All right. What are our challenges in defining a phenotype? So there are changes in diagnostic modalities over time. The example I always use is Loeys-Dietz syndrome, where you have very tortuous vessels throughout your body. That could not have been recognized before you had the ability to do either an MR angiogram or a CT angiogram, or for some reason somebody needed an angiogram. like a real one with dye injected, because if you happen to go into surgery to fix something, you'd only see that, oh, that's a funny looking vessel, but it's one vessel. You wouldn't say it was everywhere. But with these new diagnostic modalities, you can see that there's tortuosity everywhere. There are changes in diagnostic criteria over time. There are differences in medical care and medical intervention in different parts of the world, and certainly over time. And if you have a disease that's very well characterized and understood, and suddenly you have an effective treatment, and it's a completely that disorder is gone. Maybe it's replaced by things you didn't even know were there because the condition would have killed the person before they could manifest the later onset problems. Or maybe they're just normal people because you've got an effective treatment, like take congenital hypothyroidism. That's a devastating condition, but treated, you're fine. You're like a regular person walking around. Cultural considerations. Certain countries, you can't, men can't examine women. Um, Medical subspecialty bias is probably the biggest feature. So, what I always say when I give this lecture is, if a ophthalmologist have the best toys in the world, they can tell you everything in the world about your eyes, everything. But if you happen to be wearing pants and closed shoes, they might miss that you're missing a leg, right? They just might not notice. They're looking at your eyes. So, and that's the way they write their papers. They're talking about eyes. Physicians are terrible at looking at and characterizing teeth. We don't even know how to count them. So, yeah, I mean, we can count them, but we don't know what they're called. So, in any case, that's a very big problem in it for all of us who are trying to curate. Ascertainment of the phenotype at different stages of development, where somebody will report a newborn phenotype and then somebody else looking at a cohort of adults, and they don't even know they're talking about the same disease because they don't recognize that this is the development of this condition. And then, of course, phenotypic diversity at a locus, for example, the many conditions caused by mutations in lamin A, or genetic heterogeneity, seemingly identical conditions caused by many different genes. Okay, so for that, we do lumping and splitting of phenotypes with which you are all painfully familiar. And what we do, we've been splitting phenotypes on a molecular basis as long as we've had known enough genes to cause a single what we think is a single phenotype, and since the very early 1990s. So this is nothing new for OMIM. This is an example where we have sickle syndrome, and over time we've found at the moment 9 different genes that underlie sickle syndrome. And so we create the phenotypic series. This is again done by hand, and you can click here to view corresponding clinical synopses. And when you do them, you can line them up side by side to see if they're, are these really, virtually, indistinguishable, or are there really some features? And for some clinical synopses, for some phenotypic series, they really are very, very, very similar. And in some, they're a little bit more diffuse. Like, for instance, the spastic paraplegias, there's a range of age of onset and severity for those different phenotypes, but they all have spastic paraplegia. So these are some examples. And as of recently, there are 584 phenotypic series, which comprise more than 5,000 different disorders. A disorder may live in more than one phenotypic series. Okay, so probably one of your questions is, how do we curate gene phenotype relationships? So we do not do the definitive, strong, moderate, limited, refuted, disputed that you do. That's what you guys do. But what we do is decide from reading papers whether we think something's real. So we create a clear-cut gene phenotype relationship, which in your book would be limited or above. It's not disputed, it's not refuted. If there are 2 or more families with mutations in the same gene, supported by segregation, and in the case of de novo variants, a statistical test of the likelihood. Functional analysis in that situation or an animal model is a plus, but isn't required. And a number sign will precede the phenotype Mim number, and it will be added to the map. And I would say that this holds absolutely true if you have 4 heads and 3 arms. What we have more trouble with is kind of non-specific NEDs on a very broad neurodevelopmental disorders with a very broad, they range from normal with a little ADHD to people with severe. We have nothing else and no function. We may or may not put that in. We're probably a little more conservative than you guys are. We will create a provisional gene phenotype relationship if there's a single multiplex family, there's functional data from an experiment or from an animal model that supports that gene phenotype relationship. In that case, we put a number before the MIM, pound sign before the MIM number, and we add a note in the first sentence that it is one family. and the allelic variant title will say one family, and there'll be a question mark on the gene map. Okay, that means, guess what? One family. So here's an example. There's a question mark in front of this. The end of the first paragraph says one such family has been reported. There's a question mark on the gene phenotype table. On the allelic variant, it says one family. And in the gene map, it says question mark and it says one family. So I don't know how else to warn people that it's one family, but it's one family. So caveat emptor, we need some more evidence, but we believed it enough to put this in. We are not trying to add soft calls, we're trying to avoid those papers. But if by chance we go and do a deep dive into a paper that in the end we don't believe that there's enough evidence for a genotype phenotype relationship, but we've spent an hour with that paper, we would like not to read it again. So in that case, so if it's a single family, no functional data, we may add it to the gene as a variant of unknown significance. All discussion of the report will be in the allelic variant. No gene phenotype relationship will be established, and it will not be added to the gene map. In some cases where there may be more than one family or multiple reports, but we still are not sure of the validity of that relationship, it may go into the molecular genetics section of the entry, of the phenotype entry. This is an example of two variants of unknown significance, and you can see the first paragraph explains why. Okay, and then the rest of it is everything we pulled out of the paper. The second family is described, second person is described with pathogenic variants in the same gene. Then we would create the gene-phen relationship, and we would pull the phenotype information that's in this paragraph out into the phenotype entry and just leave the gene and variant information in the gene entry. Okay, so a little statistics.
Sorry, Ada, just
Yes.
to interrupt for a sec. There's a question in the chat from a few moments ago, but noting that it might be useful to list if a gene disease relationship has been validated in the GeneCC. And I know there's plans for OMIM data to be coming into GeneCC. I don't think it's going to go the other way, but did you want to give an update on where that's at?
So the ClinGen validity links and ClinGen links are in the external links of the MIM entries and the gene entries. And we're working on redoing the front end of the website and would consider ways to acknowledge whether it's been reviewed by ClinGen and provide it as a mark to a link to the ClinGen site.
Yeah, Joanna, I think Alicia's just asking whether we will be going through the GeneCC to say it would go in as supportive. And I believe there were API issues which may or may not be resolved. Joanna would know better than I would about that. The goal has always been to do that, but there were some issues with the API and maintaining forked data. So I think that's it. The plan is yes. I don't know the timing. Joanna, do you know more about timing than I do?
When I get around to it.
That was excessively honest, but okay.
And that was also great to hear about the link out to ClinGen curations as well. I didn't know that was coming in OMIM.
So we have them. We have some already, and I'm going to show the level. I think is on its way, but the rest I can show you. I'm going to show you in a minute what else we have. So this is just statistics of what's in OMIM. Let me shrink this so I can see what I'm doing here. So this is the same as if you go into statistics, you see this table. It won't be these colors. I just made it so people could read it. So again, we told you an asterisk is a gene entry, a plus. There's very few left. We're going to eventually get this to 0, but this is an entry that's actually a lumped entry with a gene and a phenotype. They're old. They're very painful to do, and they will be done. A number sign, a pound sign, I told you, is a phenotype molecular basis known. A percent sign is a phenotype molecular basis is unknown. It may or may not be mapped. The null entries are all kinds of different things. Some of them are things we're not sure are different from something else already described. Some of them we're not sure they're Mendelian. Some of them are curiosities that we like to include because why not have fun while you're working all the time? And examples of that are some particular mouse genes that may give a mouse a tail and we don't have it, or the fact that humans can't synthesize their own vitamin C. Those things are null entries in OMIM. Okay, we maintain a morbid map scorecard, which tells you the number of phenotypes for which molecular base is known, number of genes. That's the data from this morning. These are broken down further into the different kinds of classes. A non-disease is generally either a phenotypic trait, or well, it's usually a QTL. It's something like red cell size or platelet size, things like that. They're not diseases and they're not clear-cut phenotypic traits. The somatic cell genetic disease is primarily conditions that to exist in a human, you must be mosaic. Okay. And then there are a few somatic cell cancers that are there's so much literature about them. Like it's hard to not to talk about biology and not talk about the V600E mutation in BRAF. So there's certain things that are just um, um, in OMIM because there's a lot of biology and discussion about medicine and biology in OMIM, and sometimes you have to include them, but we're definitely not the resource for that. Okay, so what do we include for allelic variants? As I said, we have over 35,000, and the kinds of examples are the first mutation to be discovered, those with a high population frequency, those that cause a distinctive phenotype. So if you have two different phenotypes, you're going to have examples of the mutations that cause each of them in OMIM, those of historic significance, unusual mechanism of mutation. So, wow, that's a weird way to get that to happen, or unusual way to cause disease, or distinctive inheritance. So we would show examples of those that are dominant and those are recessive if that's, if both are associated with that particular gene. And this particular explanation is found in OMIM Help. Lots of things are in OMIM Help. Now, we only catalog select variants, as I've now told you 4 times. There are topical links to many variant resources, including population and disease-based. Inheritance resides with the phenotype. Okay, there are no dominant or recessive genes. They have variants, one from mom, one from dad. Sometimes there's copy number variants, and there's more than one, but the disease is what's inherited as a dominant or recessive trait. De novo variants are listed in OMIM as dominant because should that person reproduce, they would have a 50% chance of passing on that disorder. So it's dominant, even if it occurred de novo. Everything has to start somewhere. Examples, achondroplasia, where 80% of mutations of affected individuals are the product of a new mutation. Okay. Now, on occasion, people write things that they later find out are wrong. When that happens in the literature, we will go in and change. So and we always have the same language, so we can go find it again, and now you can too if you'd like to. So these are reclassified variants, and we just say it's been reclassified to a variant of unknown significance. We don't do any ACMG criteria for the strength of association of a variant with a disease. That we're assuming that the authors who wrote the paper are not going to tell us about things they're not sure of. Or if it's older than that, they may not have known how to do this. So this is an example of, I think this is thrombomodulin, and here it says that this was initially called something real, but now it's been reclassified based on the findings of these 2 people. And if you read further down, it'll tell you what those 2 papers had to say about why it's been reclassified. As of yesterday, there were 353 of these reclassified variants in OMIM. Okay, so these are the examples of the kind of external links from MIM entries. Not every entry will have a link to every resource, but this is just for your information. So we link to different genome resources, DNA resources, protein resources. And again, the link will only exist if there's a matching page in the other resource, and it takes you right to that page.
It's important to note that the links from a phenotype entry and the links from a gene entry are different.
Yeah. Yeah, quite different. So these are gene info. These are different variant resources that we have links to. Clinical resources, including ClinGen dosage and ClinGen validity, clinical trials, etc. So lots of links from in the clinical resources, which is why we toggle that one open for that one. Animal models, cell lines, and cell pathways are all potential links from any entry. And now I'm going to spend a little time on searching. I'm talking faster than usual. You guys are not asking enough questions. Sorry, let me add the next half of this. So in this case, I've typed in webbed neck short stature without any punctuation of any kind. And so I got, first of all, only synonyms for webbed because that's all it could give me synonyms for. And I got 6,559 entries, which could be intimidating. In this case, I used WebNeck for a proximity search. and I did an and, so I'm getting the entries that have web neck and short stature, which I've also done by proximity searching. And you'll note two things. One is now I got a synonym for web neck, Tertium coli, and I got lots of synonyms for short stature. Okay, so you can expand your search in that way. And I've got 62 entries, which is far less intimidating than 6,559. But even if you did that, and the default in OMIM is an OR search, so you're going to get a lot of retrievals if you do an OR search, but don't be intimidated. It's probably near the top of it, even if you do it in the dumb way. But the other thing is you can see that that the default search prioritizes the existence of those words in the title. And then the next level is the number of times it appears in the entry. And if I said something wrong there, please correct me, Joanna. So in this case, I have typed in dwarfism and it gave me lots of possible synonyms. And I just want to let you know that some things are hard coded. So the fact that gallstones is the same as cholelithiasis and first and kneecap and patella and English spelling versus, British English spelling versus American English spelling, all of those are hard coded back end. But some things aren't. And one thing I would tell you to be particularly careful about if you're looking is that the description of whether you're calling it intellectual disability, neurodevelopmental disorder, developmental delay, mental retardation. Remember, this is a almost 60-year-old resource. All of those things exist in OMIM. And if you want to make sure if you're for some reason trying to find all of them, make sure you pick the synonyms so that you're broadening your search.
So
Okay.
can I just
Yes,
clarify?
please.
The synonyms are hard-coded, you won't see them. They
So,
happen automatically
yes,
and
you're
you won't see
absolutely
them.
right.
So it's only thesaurus terms where we feel like somebody may consider developmental delay different from intellectual disability but others may lump them together. So it's thesaurus terms, not synonyms
Thank
that
you.
are
Sorry.
available.
Yeah, thank you for correcting me. Thank you. Thank you. Thank you. Is that any questions about what we just said? Okay. Um, so in this case, I have typed short stature and web neck, and this is 58 because it's a different time I did the search, so don't worry about that. And you can see that the matching terms show right here. Okay. And oh, this is another search thing I want to show you. This is brand new or relatively brand new. Joanna will tell you when. Imprinted regions are flagged now. So I typed in Beckwith-Wiedemann, and so I got Beckwith-Wiedemann syndrome, and I got the genes that are implicated in Beckwith-Wiedemann, and this I means it's in an imprinted region. It is not telling you that this gene specifically is imprinted. It's telling you it lands in an imprinted region. Okay. So I'm now going to transition to two things. One is I want to make sure that you know about these two tutorials. One is 4.5 minutes and one is 2.5 minutes. This one is really an excellent tutorial about how to search OMIM efficiently and effectively. The information exists in help and you can look there too, but it's only 4.5 minutes and it's really good. And then the other thing is I'm going to talk to you about Mim Match now, but there is a brief tutorial about how to use Mim Match. Okay. Now, so if you want to use Mim Match, you create an account, you're going to get an email, and you can log in and set preferences. And I'm going to show you about some of those in a second. You can follow a gene or a phenotype or a molecular series, and you can save your searches, and you can create your own custom lists. So in this case, I have gone into Mim Match, and I wanted to show you can see the pace of gene discovery for yourself. It goes back to 2000 because those are data we know are collected at roughly the same time, and these are updated daily. If this is a thing that is just driving your life, please go look. And then I don't know why this is exactly the same, but in any case, this shows you the different components of Mim Match. You can ask to be notified when a new gene phenotype relationship is cataloged in OMIM. So the good news is you will never get more than one email a day, and that email will tell you about anything you happen to care about. And if there's nothing new that you happen to care about that appears, you're not going to get an email. So we're not going to waste your time. But if you want to know about new gene phenes, you can do that. If you're following particular genes or phenotypes, you can pick those here. You can pick to follow phenotypic series, and currently 584 of them, comprising 5,000 disorders. You can pick to be notified about a new gene phenotype relationship that is, sorry, that's wrong. Oh my God, I'm so sorry. It's now quiet. You can pick to know about new phenotypic series. You can, if your particular phenotypic series you're following, you can ask to be notified if something is new or removed from it, or if there's any updates to entries within it. Again, never more than one email a day. You can create your own lists. So, and you can either start with diseases or start with phenotypes. We can start with genes. In this case, we're picking genes, and we are going to get this table and put them in, and you create, this is like table one of your paper if you're writing a review or if you need to organize information. So, and this is organized. OMIM defaults to organizing by location, but you can organize in other ways if you'd like. The other way you can put it in is you can put phenotype numbers, and then you can again create it the other way around. So these are this is another table, and if you want to follow entries within this, you can just make sure you're picking all of them in your in Mim Match to continue to follow. Now, this is a relatively new feature of OMIM, and it's currently only available in Mim Match. It will someday go on Mother Mim, but Joanna will tell us when, or we'll decide when. She may not be able to tell you today. And these are tabular lists of phenotypes brought together by a shared mechanism, etiology, or pathology. Currently, there are 16 molecular series, and they have 316 or so members. And this is the list of what they are. You can go into each of these and see them. And this is an example of the non-protein coding morbid genes. So these are RNA genes. There's 19 genes that cause 26 different conditions. So you can see there's 2 different conditions caused by this one and so on. There are 11 are autosomal recessive, 14 are autosomal dominant, 1 has both. I just kind of looked at what's different or the same about them. 15 have no effect on growth, 1 has overgrowth, 10 have short stature. And 15 of them have some sort of neurological involvement, including developmental delay, intellectual disability, ataxia, seizures, brain anomalies, etc. But it's a remarkably broad phenotypic spectrum. And of course, this is really quite a new series. So this will expand over time. And the ability to even detect this is new. There's a molecular series for repeat expansions and a molecular series for alanine tract expansions. And when I looked at these, I was really surprised to see that most of the non-alanine repeat expansions are predominantly a neurological phenotype or neuromuscular, and most of them are progressive. They get worse with time, whereas the alanine tract expansion disorders are often static congenital anomalies. Only a few of them have, brain or, cognitive involvement, and they tend to be static. They're not progressive. So I just found that really interesting. And it lets you look at things in a way you might not have thought of before. Based on this paper published in, I think, in 24, we have pulled out these different disorders of the epigenetic machinery. And so people can see them together in molecular series. So each component is in there, and you can look at it. And in a condition can sit in multiple phenotypic series. It can also sit in a phenotypic series and a molecular series. So in this particular example, this is spastic paraplegia 93, caused by recessive mutations in the NFU1 gene, and it sits in the spastic paraplegia series where you would expect it to, but it happens to be a disorder of iron sulfur biogenesis, and so it lives in that molecular series as well. And in this case, this is an example of a gene phen graphic taken from within Mim Match, which shows that in this case, I have closed spastic paraplegia and open multiple mitochondrial dysfunction syndrome. This is starting from spastic paraplegia NFU1, and you can see these phenotypes and that it's part of, this is the bright pink shows you that this is a molecular series. And then if I go the other way, in this case, I've closed the mitochondrial the multiple mitochondrial dysfunction syndrome, and I've opened the spastic paraplegias. There's still too many of them for you to read, which tells you there's a lot of spastic paraplegias. This is a couple of others. This is the morbid genomic regulatory sequences. So these are genomic elements. They're not RNA genes, non-coding RNA genes. These are genomic elements. So they're enhancers, repressors. They don't create an RNA or a code for a protein. Their disruption or dislocation creates a recognizable phenotype. And so far, there are 10 and maybe 11, I think it's 10, members of this series. And they're remarkably variable as well. And some are very far from the gene they affect, and some are nearby. But we place them, if they're not in an intergenic region, we place them within the gene that they on the genomic coordinates that they are found. We don't put them on the gene that they affect. So the example of this, I think there's two of them. The lactase persistence is pretty far from the lactase gene, but it's in this one. And the other good example is the one that the ZRS, which controls sonic hedgehog. I think it's like 2 megabases away from it, but it controls its expression. And these are real curiosities of life that we put together so we can find them again. These are the positional effects of structural variation. So these are structural variants that brought one part of the genome next to another part of the genome. It doesn't even have to be on the same chromosome. There can be anywhere. Some of them are nearby and it's disrupted a tad, but some of them are, something from chromosome 12 went in front of something in front of chromosome 17 and create recognizable recurrent phenotypes. And this is called Liebenberg syndrome, and the bones and joints of the arms look more like the legs, the feet, the lower extremities, whatever words I'm supposed to be using. This is Endo syndrome. It's a limb-only phenotype, and these are really unusual bones right here. And in this case, this is congenital hypertrichosis, and things have been rearranged so that you're expressing lots of hair in places that you would not normally have it. And now I'm going to transition for a minute to functional and useful ways to search OMIM. So if you are doing a phenotype search, use the fewest terms and build out. Avoid using, it doesn't matter, but it won't help you to use words like syndrome, disease, etc. Use Marfan, not Marfan syndrome. Use Noonan, not Noonan syndrome. And there are example searches on the homepage that will help you see that. Fielded searching is very powerful. Joanna is the wizard at this, but it's possible to do fielded searches of everything within OMIM. And there's detailed instructions on how to do it from search help on the help menu it's the top of it, on the top of the menu bar. And there are advanced search pages for OMIM, for its gene map, for clinical synopses, and they're available under options in the options tab on the gene map searching. So one of the other things that I want to be sure I show you, and Joanna, feel free to hop in if I'm not doing this right, is in this case, I've done a standard OMIM search for the word hyperplexia, the hardest word in English to say, in my opinion. And I have 24 retrievals. If I click this gene map, I previously showed you what happened if you click clinical synopsis. If I click the gene map, I see those 15 entries and where they are. And if I click on well, these are phenotypes only because I started from a phenotype, and I can see where they are and whether they're on the same chromosome, different chromosomes, etc. And I can, if there are allelic disorders, this is an example of this gene has, this gene, mutations in the gene cause both of these disorders. And then the other way is I've now put, I haven't, Joanna has put in title, which restricts the search to, it has to be in the title, zinc finger, okay? And so she's got 620 entries that are zinc fingers. Okay? That's a lot of them. If you want to see where they are and what they cause, you click the gene map table. It'll take you to the gene map, and then if you click phenotype only, it restricts it to the zinc fingers that have a known phenotype. And in this case, you can see it's 103 of them. And you can look over them quickly. So again, it's another way to toggle between phenotypes and genes and sort of get the lay of the land of where you've landed. There are tutorials and guides available through search help under the help menu, the quick reference guide, which is the same as our brochure if you've ever run into us at meetings. That's available to download under FAQ 1.10. There are video tutorials available on the homepage. I already told you about those. And then there's a current protocols that's a couple years old, so there may not, some of the newest things may not be in there, but it's a very detailed mechanism to search OMIM. And to just show you what we have, then how we link within OMIM. So these are topical links to useful resources that you guys use every day. But you can mouse, so if you don't know what something is, you can mouse over it to get a definition. So I've moused over gene reviews, and that defines it here. As you all know, you always want to know when something was last updated. In this case, if you look at the bottom of a MIM entry, it tells you the creation date and contributors. If we click on that, it shows you that this was last updated on this date and that some edit was done by Anne Lopez Stumpf on this date. And by the way, Everett Noonan syndrome precedes 1986. I'm sure it was in 1966, but when OMIM was integrated into or made available for download, which is in 1986, not for download, for dial-up is what we were doing in those days. It was before the World Wide Web. That was the date that everything went from the book to being on the internet. So that's why it looks like it's later than it should be. It is. If you go into gene reviews, you can see this was last updated in February of 2022. You all know there are 912 gene reviews. They are expert authors. They follow a standard format, which is very useful, and they're updated every 4 to 5 years with an official rewrite that is peer-reviewed, but they're also updated as needed by the staff of gene reviews to make sure if there's any new information that gets included. This is, I just want to show you that ClinGen resources are integrated. So in this case, this is the PTPN11. You can see there's a ClinGen dosage entry and a ClinGen gene validity entry. And if we click on it, this is the dosage sensitivity, and it takes you to the page for information on PTPN's gene disease validity information. You can click on that information if you're interested. You all know what ClinGen is, so I'll skip that slide. And then I don't know how many of you are familiar with GeneScout. It might be useful for you in the work you're doing, so I want to spend a second to tell you about it. It's genescout.omim.org, and it is built on OMIM. So it changes daily. If you do a search today, it might have different information than what you'll see tomorrow. There is a very, very useful tutorial. It's a little longer because it's a little bit harder not harder, but it has a lot of information. So it's a 5-minute tutorial. So you can put in a list of coordinates that you're interested in looking for. Maybe it's a deletion, maybe it's regions of homozygosity in a patient with a problem from a consanguineous family. And you can put the coordinates in here. You can also add a second person or a second list of coordinates if you're interested. And you can either look at what is shared or what is not shared. So for instance, if you have an affected proband and an unaffected sibling, you can see, well, what's unique to this person to try to figure out what it is. You have to designate the build that the information comes from. And you get a retrieval. Well, here I'm going to compare two people. And I am going to, and what I get back from this is I get back that there are, so all genes is all RefSeq genes. OMIM is all OMIM genes. So they're not everything in RefSeq is in OMIM. And there are 354 OMIM disease genes, of which 139 dominant, 236 recessive. So I can look at this and I can compare side-by-side clinical synopses directly from this retrieval. And I can also, if there are any of these links, it will be in here and you'll be able to go directly to it. Okay. And then the other thing is if I got too many retrievals and I'm like, okay, I don't want to go through that many, I can limit it by some of the phenotypic features of the patient. So this person has microcephaly and seizures, and now I have only 39, and I can start to look through them. And again, I can compare selected by clicking over here. All right. And I think I'm actually done. So I'm going to stop sharing And I'm going to look at all you beautiful people, but basically we're looking at each other. And I am happy to answer any questions, to go live, to, whatever you need to know. That's what I'm here for. We have 2 whole minutes. Is there a question it may be useful to list if that gene disease is validated in ClinGen GeneScout? I think we talked about that before. Okay.
That was perfectly timed. So we only have a minute left now. If there's not been a burning question, we might wrap up there. But I want to say a huge thank you, Ada, for what was an excellent whirlwind overview, but it needed to be a whirlwind because there's so many features in this.
Yeah.
What really is an invaluable resource. So a huge thank you to you and the whole team that maintains it every day and keeps adding new features. I feel every time I hear you speak about the database, I learn something new about what it can do. So
You know
thank
what? Every time I walk into Joanna's office, I learn something new. When she comes into mine, she learns something new because there's
What I know is that we all approach the information differently.
Yeah.
And so there's different ways to get at it.
Yeah. So thank you very much. It was a pleasure, and I will see some or all of you soon. Take care.
Thank you.
you so much.
Thank
And we will see you all again on June 11 for our next meeting. It's going to be more of a ClinGen focus call where we're going through some of our updates to our variant curation procedures. So we will see you again in June.
Thank
Thank
you.
you
Take
all.
care.
Bye.
Bye.
Bye.